Okay, there are times when the cell has to bring molecules into or out of the cell against a gradient. And so, in order for this to be possible, the cell has to put energy into the process. So, whenever the cell is forcing molecules against the gradient, meaning moving them from where they're already low towards where they're already high concentration, that's called moving it against the gradient, or sometimes we say moving it up the gradient. And in order to do that, energy has to be used. So we said that diffusion is a passive process. It happens on its own. It doesn't require energy. And that moves things from a higher concentration area to a lower. But active transport does exist. And there are particular examples of active transport that are typically shown for any starting biology student. And one of them is shown here. It's called the sodium-potassium pump. It's going to actually move two things across the membrane, sodium ions and potassium ions. Potassium has the symbol K. Oops. And what's going to do is it's a carrier, so it's going to change shape, because you remember carrier proteins move things across membranes and change shape. And the sodium is going to move against the sodium gradient, and the potassium is going to move against the potassium gradient, and energy is used to make this happen. And so a lot of considerations here are happening all at once. It's a carrier protein because it's changing shape, and these ions are being moved against their gradient. You would have to be told what the gradients are so that you would know that, but I'm telling you for this one, it is against the gradient. And what it does is it requires energy in the process because it doesn't naturally happen that way. So the cell has to put in energy. And so if you were shown the gradient, what you would see here is that there's more sodium in the extracellular space. And what you're seeing in this kind of first frame right here is that the sodium is going to be moving from the inside of the cell towards the outside. So it's going to increase an already high concentration outside the cell. So that's moving against the gradient. And so energy has to be used to make that happen. Then when that sodium is released out of the cell, what's going to happen is two potassiums come in and they're moved into the cell, but there's already a higher concentration of potassium inside the cell. And so both the sodium is moving against its gradient, which happens to be higher on the outside of the cell than on the inside, and the potassium is moving against its gradient because there's more potassium inside the cell than there is outside. And so both of them being moved across their gradient, it doesn't have to be two things that are moving, but the sodium-potassium pump happens to be two different things moving through the same carrier, which you'll remember is possible. Usually one thing moves, but it can be two. And they're being moved with the help of an expenditure of energy. So it fits the definition of active transport. So that's called the sodium-potassium pump. It's a good, well-studied pump that, and we use the word pump, it implies that there's energy being used when you use that term. But it's a channel protein, a carrier protein that requires energy to do its function. So you have, like I said, if molecules are being moved in the direction of the gradient through a channel or a carrier protein, it's called facilitated diffusion. If molecules or ions are being moved against a gradient with a channel protein or a carrier, it's called active transport. And so those are terms that can be used. Other terms, though, that are also possible when we look at a different aspect of the transport event, we can call the carrier proteins by these terms: uniporter, symporter, or antiporter. A uniporter would be a carrier or a channel protein that moves one molecule at a time in one direction. So single, one, transport. A symporter moves two molecules in the same direction. And an antiporter moves two molecules, but they move in opposite directions. And so I'm going to go back to this example. This is an antiporter because the sodium is being moved out ultimately, and the potassium is being moved in through the same channel protein. So that fits the definition of antiporter. And then kind of a separate consideration is that because both of them are moving against their gradient, it's also an active transport. So it's both. All right, let me go back a little further and let me see if I can find another one. All right, this one, because only one molecule is being moved in one direction, this is an example of a uniporter. And since in this example, the arrow of movement goes with the gradient from higher to lower concentration, it's also a facilitated diffusion. So you're combining the two terms based on looking at different aspects of the event. So facilitated diffusion and active transport, focusing on whether or not the movement of the particle or molecule is going with the gradient or against the gradient. And then uniporter, symporter, and antiporter has a different aspect that it's looking at. Let me flip back here. Whether the molecules are being moved just one, two in the same direction, or two in opposite directions. So this is a whole separate consideration from this. So you have to look at different aspects of the example, and then you apply the appropriate terms. So that is the end of our discussion of facilitated transport, meaning using these pore proteins, these channel proteins or carriers, to allow things to move through in a selective way. Only monomers or things that are smaller like water molecules can move through these channel or carrier proteins. If there are larger molecules, they have to be brought in by a vesicle. And sometimes we call it a vacuole. But ultimately, what a vesicle or a vacuole is a compartment in a cell that is surrounded by membrane. And the membrane is just holding some kind of material. So in this case, what we'll be talking about are two processes, endocytosis and exocytosis. We've already talked about these to some extent. I'm just going to connect this to what you've seen in a previous chapter. Endocytosis means moving substances into the cell by a vesicle or a vacuole. And how this happens, I'll show you on the next slide. This is the artist showing endocytosis. So let's look at this one here, for example. On the left side is an artist drawing a sequence of events. Here's 1, 2, 3, 4, 5, kind of a time lapse. And on the right side is an actual microscopic image of this happening. It's sort of a freeze frame. And so some kind of large molecule or even a whole cell can be taken in. A bacterial cell could be taken into a eukaryotic cell, for example. When the cell or the large food molecule, as it could be, gets to the outside surface of the plasma membrane sort of sinks in and starts to surround that molecule or that particle. And it keeps going around until the edges touch together, and ultimately a vesicle pinches off and moves into the cell. So that would be a food vacuole or vesicle. And that is called phagocytosis. And you might recall phagocytosis was the way that molecules are moved into the cell, and then that food vacuole fuses with a lysosome for the process of intracellular digestion to break down that macromolecule into the monomer units inside the cell. So phagocytosis is one that I want you to focus on. All right. Let me go back. All right, so we're going to put a star by phagocytosis. Pinocytosis and receptive mediator endocytosis are just variants, just slightly different variations of this same concept. You know how biologists have to put more vocabulary words on every tiny version of the same process. So these are processes that take in materials by vesicle. The opposite is exocytosis. Exocytosis is releasing something from a cell. When a vesicle moves inside the cell, it moves up to the cell membrane, it fuses to the cell membrane, and then it pushes the contents of that vesicle out. And so we discussed exocytosis, particularly when we talked about protein secretion. The proteins were in a vesicle. As they were moved from the Golgi, the vesicle moves to the plasma membrane, it fuses, and that dumps those proteins out. So we've touched on these already, but they are specific examples of these more general processes. So there are other times when endocytosis happens other than just intracellular digestion, but it's still a process of bringing something in. Exocytosis is releasing something out, and there's always a vesicle or vacuole that's involved with this transport because the items that are being moved across the membrane are too large to go through a channel protein or a carrier protein. All right, and this picture is exocytosis. So we'll put a star on this one. Just a concept, the vesicle coming from, in my example for protein secretion, coming from the Golgi, and then touches the membrane, and then the membranes fuse, and that creates an opening, and the contents of the vesicle are dumped out. And this is an actual micrograph, and they colorized it to match the artist picture with this blue and green coloring for whatever it is that's being released. So, but that's exocytosis, the release of something that the cell made, or it could be wastes. If the cell made something on purpose, it's called secretion. If the cell is releasing waste, we usually call it excretion, cellular excretion, but it's a release of something from the cell by vesicle.